In this video, we are going to talk about microscopical evaluation in detail. In the previous lecture, we had studied about the different types of drug evaluation. That is, we had enlisted it physical, chemical, biological, and your microscopical evaluation. So here we are going to study about the microscope, the various agents that are used as a chemomicroscopic reagent or clearing agents. Then we'll look forward for quantitative microscopy, micrometer, and camera lucida. Moving ahead with the microscope, as we are all well aware that it is an optical instrument which comprises of lens or combination of lens that is used for the magnification of minute or small object. दो टाइप के होते हैं, सिंपल माइक्रोस्कोप, कंपाउंड माइक्रोस्कोप. सिंपल में वन सेट ऑफ लेंस विद लोअर मैग्निफिकेशन, कंपाउंड में टू सेट्स होते हैं लेंसेज के, प्लस यू हैव आई पीस ऑब्जेक्टिव with higher magnification. Okay, this is the diagram or picture of simple microscope. It has one lens. It consists of the support system, that is your base, body tube, folder arm, okay, stage, and then you have illumination system, you have mirrors, then you have certain adjustment screws and clips. Now we have the recently used compound microscope which has objectives. Okay, it shows higher magnification. It consists of objective, eyepiece, iris diaphragm, all right, and external source of light या फिर कोई भी external source of light, it can be either your sunlight or certain tube light source of light. Now here you have three objectives, 45x, 100x, and 10x, all right. And again you have the folder arm, the body stage, and the body tube as the supporting system. And in illumination you have mirror, then you can have iris diaphragm, tube light, external sources, okay, of light. Then you have magnification system in which there will be lenses. Magnification system में क्या आएगा? Lenses, all right. Let's move ahead. As I said, it consists of three major systems. Support system में you have body, stage, and body tube. Illumination में you have light source, mirror, iris diaphragm, condenser. Magnification में set of lenses जो object को magnify करने में काम आएगा. Okay. Then we have different illuminating systems used in microscope, जैसे कि plane mirror, concave mirror, substage, interchangeable mirror, built-in substage lamp. Now if you see here, you have a built-in substage condenser, right? But कुछ जो होते हैं वो interchangeable होते हैं. Then light adjustment में you have condenser and diaphragm. जो नीचे से open होता है, that is your diaphragm. Now let's move ahead to the recent advances in microscope. With the time being, there are innumerous microscopes are being invented, such as you have monocular, binocular, dual viewing microscope, trinocular, projection microscope, fluorescence, UV, electrolyte. This all you can study in detail while surfing on net. Okay, just for your extra knowledge, I have enlisted here, and these types of microscope are very valuable and are used to study of extreme research. All right, now we have certain clearing agents. Now in order to clean the microscope properly, we use chloral hydrate solution, hydrochloric acid, organic solvents like ether, oily cleaning reagents. Okay, this is majority of times it is asked in your MCQs, what are the cleaning agents, and also in your Viva was. Okay, that is your chloral hydral solution, hydrochloric acid, organic solvents like ether, oily cleaning reagents. Now we move to a very important topic, that is microscopical measurements. The dimension of cell, that is your fiber, stone cell, or trichomestic cell contents such as your starch grains, pollen grains, etc. of crude drugs, both in entire and powder form, can be measured using two instruments or two apparatus, that is your micrometer, which is micrometry, and your camera lucida. Now micrometry is a scale which is used to measure the microscopic objects. It is expressed in microns. You can see micrometer जो होता है ये एक ऐसी स्केल है जो माइक्रोस्कोपिक ऑब्जेक्ट्स को हम लोग स्टडी करते हैं, ये माइक्रॉन्स में जो है एक्सप्रेस होती है. It is a micrometric evaluation which is useful for the study of dimension of starch, grain, calcium oxalate crystals, fiber, oil cells, other cells, and cell contents in powder or ungrounded crude drugs. The size of an object part can be measured with help of two mirror micrometer, that is stage micrometer and eyepiece micrometer. Eyepiece micrometer is also known as ocular micrometer. माइक्रोमेट्रिक स्टडी होती है माइक्रोस्कोपिक ऑब्जेक्ट्स की जो माइक्रॉन्स में एक्सप्रेस होती है. इवैल्यूएशन ऑफ स्टार्च, ग्रेन, कैल्शियम ऑक्सैलेट क्रिस्टल्स, फाइबर्स, ऑयल सेल्स, अदर सेल्स कंटेंट्स एंड अनग्राउंडेड क्रूड ड्रग्स के लिए यूज होता है. द साइज ऑफ एन ऑब्जेक्ट और पार्ट ऑफ इट कैन बी मेजर्ड यूजिंग the stage micrometer or the eyepiece or the micrometer, that is ocular. You can see stage micrometer ऐसा होता है in which one division is equal to 0.01 millimeters. और यहाँ जो होती है that is a scale present. ठीक है, ये stage micrometer है which is used in micrometry for the evaluation of your dimensions, okay, of various organized crude drugs. You can see I have just enlarged for you all. There is a scale, as I said, one division is equal to 0.01 mm होता है. Alright, from it is 0 to 10, this is the stage micrometer. Okay, whenever you are seeing in a microscope, it will appear like this in the magnified image. Then you have ocular or eyepiece micrometer, okay, which is to be placed in such a way. It is of this kind. Okay, you have to first open the bracket, then in the bracket you have to at the end of the eyepiece just remove it and insert the ocular micrometer facing with the printed side facing downward. The printed side should be faced downwards. Alright, so that were the two parts of the micrometer which is used for measurement. You can see here I have shown you the stage micrometer here. Okay, the stage micrometer and ocular micrometer should calibrate or coincide with each other in order to find the dimensions, right? You can see here how it is coinciding. You can see यहाँ पर that's not proper, but यहाँ पे you can see certain lines are coinciding here. So that is the reason this is your stage micrometer, this is your ocular micrometer, so they both are comparatively studied and understood. Okay, calculations you can see here. One division of stage micrometer is 0.01 mm, that is your 10 mu. Okay, 10 microns. If 5 division of eyepiece micrometer is equal to 3 division of stage micrometer, then the value of 1 division of eyepiece micrometer is calculated. अब यहाँ पर क्या 1 division of stage micrometer is equal to 0.01, ये हमारा स्टैंडर्ड है. ठीक है, अब सपोज यहाँ पर जो है हमारा 5 और 3, ठीक है, यहाँ का 5 और यहाँ का 3, यू कैन सी अगर यहाँ का 5 और यहाँ का 3 मेरा कोइंसाइड हो रहा है तो मैंने यहाँ वही लिखी कि 5 ऑफ stage micrometer, so 5 of ocular micrometer and 3 of stage micrometer. ठीक है, अब इज इक्वल टू डाला है, मतलब जितना 5 डिविजन्स का रहेगा तो जितना 3 डिविजन्स का रहेगा उतना ही 5 डिविजन्स का रहेगा. तो अगर 3 डिविजन्स का अगर 1 डिविजन का 0.01 है तो 3 डिविजन्स का 0.03 रहेगा. एंड एज इट इज़ इन माइक्रोन्स आई हैव टू कन्वर्ट इट इनटू माइक्रोमीटर, इसलिए मैंने 10 से मल्टीप्लाई की. ठीक है, अब 10 से मल्टीप्लाई करने के बाद मुझे क्या मिलेगा, यहाँ पर जो मेरा 3 डिवीजन रहेगा वही 1 ऑक्यूलर रहेगा. फिर वो 1 ऑक्यूलर को कैसे ढूंढेंगे, x से. सो विल क्रॉस मल्टीप्लाई एंड विल गेट द लीस्ट काउंट. ठीक है। अब जो भी लीस्ट काउंट होगा उसका हम लोगों को जो भी क्रिस्टल होगा उसका डायमेंशंस पहले निकालना है एंड मल्टीप्लाई बाय 100 करना है, ठीक है। देन यू हैव टू सी हाउ मेनी लाइन्स आर रिक्वायर्ड, देन मल्टीप्लाई द नंबर विद लीस्ट काउंट. Alright. Now we have the another one and used in the microscopical evaluation, that is your camera lucida. Now the camera lucida or drawing ocular is useful for tracing a magnified image of object under microscopical study with the proper adjustments of camera lucida and illumination. अब camera lucida एक ऐसा इंस्ट्रूमेंट होता है, ऐसा दिखता है जो कि हम लोग अटैच करते हैं एट द टॉप ऑफ द आई पीस, और इन सच अ वे एक ये ऑक्यूलर डिवाइस है कि वेन एवर वी आर ट्रेसिंग दैट ऑन द पेपर, okay, it is your hand can be seen through that camera lucida, ठीक है. You know, just tracing of a magnified image, like you are seeing that image in this, you are seeing the image from this. यहाँ पर आई पीस से अटैच करते हैं, फिक्स करते हैं आई पीस को, और यहाँ से पैरेलल होना चाहिए वो आई पीस को. सो यहाँ से देखते, तो जब यहाँ से देखते हैं तो वो जो भी इमेज होगी माइक्रोस मतलब ग्लास लाइट पे वो मैग्नीफाइड दिखती है, एंड थ्रू दिस कैमरा लूसिडा वी कैन ड्रॉ इट, and you know you can see your hand working. Okay, you will see the magnified image but your hand will be seen drawn on the paper. Okay, दो है, एक swift iris है which is like this, which we used in our laboratories, and this is Abby's model which consists of the mirror. Okay, see here how to install camera lucida. This is your eyepiece. Here you will just first remove this, okay, and just fix it tightly, and just place this you know camera lucida just parallel to the eyepiece. Okay, and this is your Abby's model which consists of your mirror. This is the ray diagram, like this is the eyepiece you are looking from here. So the observer is when looking, you know, the angle is drawn in such a way that his hand is seen in the camera lucida but he is working on the paper. Okay, microscopical evaluation we will be studying in the next video. We'll just enlist what are microscopical evaluation. We have calcium oxalate crystals, starch grains, pollen grains, trichomes, leaf surface constants, tomatal numbers, tomatal index, Beliside ratio, win islet number, wine termination number, and Lycopodium spore method. I hope you have understood the basic introduction of microscopical evaluations. Thank you and subscribe my channel.